we have discussed how good is the link between neural events and vascular events, right? We have seen that the vascular response scales proportionally to the intensity of the neural activation and scales proportionally to the timeline of the activation. So the next question I want to address is how good is the link between neural activities and bold response? Now, the first aspect of this link is to try to understand what exactly does bold reflect? What is it? What part of neural activity is bold mostly connected to? Now, to look at this, we're going to look at an experiment by Nico Logothetis' group in 2001. Now, they had anesthetized monkey in a 7 Tesla MRI machine. And what they did is they had an electrode to record the neural activity implanted in visual cortex. And at the same time, and you can see down here the area that is more or less recorded by the electrode. And then you can also see the parts of the brain that responded, the activation in response to stimulation, the bold activation in response to stimulation. And this was a visual type stimulation. So again, the question is, what aspect of neural activity does bold correlate the most with? And we're going to look at 3 different aspects. We're going to look at bold, which I will show in orange. Then we're going to look at single units activity. So this is single neurons action potentials. Then we're going to look at multi-unit activity, so groups of neurons firing action potentials. And then we're going to look at local field potentials. Local field potentials pretty much are dendritic activity. So this allows us to see whether bold correlates the most with action potentials or dendritic activity. So here, this is time. Each line matches one of these events. So for example, you can see, so this is when the neural stimulation started, the visual stimulation started. This is when it ended. And what you're seeing under in the background, the orange full line, that is the bold signal. Okay. Now the question is, which of the other lines correlates the most with bold? And you can see single unit activity in blue. So as soon as the stimulus starts, you get this big increase in single and multi-unit activity. But then it kind of goes back down to baseline and stays at baseline. Conversely, local field potentials get this giant increase at first and then remain elevated throughout the period of the stimulation. And then once stimulation turns off, local field potentials also turn off. So if you correlate these 4 signals, it turns out that the biggest correlation is between bold and local field potentials. And if you change the timing of the activation, for example, if you make the stimulation, shorter in time, you do see that the multi-unit activity doesn't change that much. It's a big spike at the beginning, and so is the single unit activity, and then it goes back to baseline. Whereas local field potentials, again, scale with the stimulation, and so does bold. And finally, you can make it even shorter, and you still see the same thing. So overall, it looks like bold is mostly correlates with local field potentials. So it is possible that bold might capture to a greater extent the dendritic activity around the synapse and around the synaptic cleft. And that kind of activity might be more tightly linked to bold than action potentials. Now, an important aspect of the question of how good is the link between neurons and bold is the spatial localization and the timing of events. Now, there's a wonderful experiment in which they looked at the distribution of neural activity and bold activity. So it's in this experiment, they recorded with electrodes over cortex neural responses, and then they recorded at the same time neural events with bold. So the site of stimulation, so they would stimulate the snout, the tongue, the chin of the animal, and then they would stimulate different places on the hand. And what you're seeing here is a diagram of the so-called precentral sulcus, so the motor part, and the post-central sulcus, which is the sensory part of the brain. But what matters is that what you're seeing, so each little dot, see, each little dot is where an electrode was recording. And then when you see this full, for example, this full pink area, these are all the electrodes that responded to stimulation of the hand. Okay, so the electrodes sensing directly neural activity, those that responded to hand stimulation are those inside the pink shaded area. Similarly, the green shaded areas here, here, and here, these are the parts of the brain that responded to stimulation to the chin, mouth, and tongue. So this is, if you can record directly from the neurons, these are the neurons that were active. So now the question is, how similar is the pattern, spatially speaking, is the pattern of activation of BOLD? And that you can see with the big dashed areas. So for example, this big dashed area right here are the areas of increased BOLD signal upon hand stimulation. And there's another big one here. And you can see that overall, for example, this big area of BOLD activation, of BOLD response, matches pretty well this area of neural activity. And even though slightly warped, the same is true here. We know that here there's real neural activity, and we know this from electrodes recording the neurons with electrophysiology, so recording changes in electric fields generated by the neurons. And you can see this big area of BOLD activation. So overall, the two are co-localized pretty well. More or less the same is true for stimulation of the mouth and tongue. So you can see we said this, this, and this, and actually this one down here too, are areas that we know in their neurons increased their firing rates and their activity. And BOLD gave a big response in this big green dashed area. Notice there was no noticeable BOLD over this area here. So do the two co-localize? Well, yeah, pretty well, at least sort of coarsely. For example, here, we know neurons are active in here, and the BOLD response is pretty well co-localized. The same is true here. And here too, we know that these neurons were active, these neurons were active, and we see a general increase in BOLD signal nearby it, proximal to it. Of course, very noticeably, the two are not perfectly aligned. And you can see it very well here, right? The area that is activated according to BOLD is slightly different, is overlapping, but slightly different with the area that is neurally active as recorded with electrodes. So are they co-localized? Yeah, more or less, not perfectly though. So BOLD is not a perfect reflection of which neurons or which parts of the brain have increased neural activity. So this is with respect to spatial localization. With respect to timing, right, we can also look at whether the timing of mental events matches or is somehow reflected in the timing of the BOLD response. So to do this, a classic experiment is to use the mental rotation paradigm. Now, if you haven't already encountered this, the mental rotation paradigm is a paradigm where you're shown two three-dimensional figures, just such as these two. And you're asked, think over whether it's the same three-dimensional object just rotated or whether it's two different objects. And there's a wealth of psychological research to show that the further apart the two shapes are rotated, the longer it takes to realize whether they're the same or not. Almost as if you were seeing the two objects and rotating one into the other. It turns out, for example, let's say that you have two trials, one where they are very nearby, so it's easy to check that they are the same object, and one where you have to rotate one object more to see if it matches the other one. So the trial where the two objects are nearby, you don't have to rotate them more. Trial A, typically on average, gives rise to a shorter bold response than, and a faster, so maybe you respond after 4 seconds, and so you get the blue bold response. Instead, in the other trial, maybe you take 5.5 seconds to answer, and you do get a slightly longer bold response. So here too, we see that the timing of neural activity is to some extent at least proportionally represented in the timing of the bold response. So now we know that the bold response does preserve to some extent the spatial localization of neural activity, if imperfectly, and does preserve some aspect of the timing of neural activity. And this graph at the bottom just shows over a number of different trials, the length of each trial, which is on the x-axis, is proportional, or rather, the length of the bold response, how long it lasts on the y-axis, is proportional to how long the response time, so to the length of the mental event and presumably of the underlying neural activity. Now here's one example from a paper from De Pisapia and Braver. They used a very clever paradigm. They were trying to understand what is the role of BA10, an area right in the front of frontal cortex. And their paradigm went more or less as follows. They would give you a number, for example, 6, and they'd say, hold this one in mind. Then they'd say, okay, 4, and then times 2. So you would have to take the 4, multiply it times 2, 8, and then say, whatever is the result of that, minus 3. And so you'd say 8 minus 3, 5. And then there would be the integration step. This is when you take 5, whatever number is the last number you calculated, and add it to the number you were holding in mind from the beginning. And so, for example, here you said 4 times 2, 8 minus 3, 5, integration plus 6, 11. And then you'd keep going. Okay. Now, their paradigm changed the position where the integration happened. Sometimes they would integrate it here. So, for example, you do 4, and then they would say, okay, integrate here. So you do 4 plus 6. Sometimes it would be here. So you do 4 times 2, integrate. Right? And so it'd be here. And sometimes it'd be here. So we can tell apart. So there are 3 types of trials. One have an early integration, one have a middle integration, and one has a late integration. And so they looked on average when the bold response in BA10, the area of interest, would peak in the 3 trials. And it turns out that if you look at the integration trials, the early integration, the green line, so when you didn't integrate it right here, peaks first. BA10 peaks right here and then falls off. When you integrate in the second integration step, so when the integration happens here, BA10 remains active, and then integration happens right here and then falls off. When the integration is late, BA10 remains active and stays active and stays active. The integration happens here, and then it falls off. So this to them was evidence that BA10 served an integration purpose. To us, it's interesting because it's a nice proof that the timing of mental events is reflected in the timing of both. If the integration happens here, you see the bold response peak here and then fall off. If the integration happens here, you see the bold response peak here and then fall off. If the bold response happens here, you see the activation peak here and then fall off. So this is a wonderful demonstration of the fact that there is a proportionality between the timing of mental events, thus the timing of neural events, and the timing of the bold response. So the link between the two is pretty good.